right, well, we will get started. So welcome everyone. We're really thrilled that you're here. This is the Field Biosecurity 101 webinar, and it is hosted in partnership with the Partners of Amphibian and Reptile Conservation with our Disease Task Team and the Association of Fish and Wildlife Agencies. My name is Carrie Wickstead, and I'm the State Agency's Coordinator for PARC. And with me today are members of our Disease Task Team who will be presenting, as well as a few others who will be assisting with answering questions and helping with some of the resources in the chat. So we're all so thrilled to see so much interest in this extremely important topic. And this is the second webinar in our Disease Task Team series. For those of you not familiar with PARC, Partners in Amphibian and Reptile Conservation is an inclusive partnership. And we include folks from state, federal, non-government organizations, industry, and all of the above, people who have interests in reptiles and amphibians, educators, anyone who wants to join in. And we have three main strategic areas of focus. We have network growth, species conservation, and habitat conservation. And PARC would not exist without our fiscal sponsor, the Amphibian and Reptile Conservancy. So this webinar is thanks to them as well as other partners. Just to give you an idea of some of the products that come out of PARC, just last year, the Timber Rattlesnake Conservation Action Plan was published. So you can find that on our website, which I will drop in the chat in a second. We also have some national task teams like the Disease Task Team, which will be featured here today for today's webinar. They help run our herp disease alert system, which we'll hear about a little bit. They help put out fact sheets. So we have 8 new disease fact sheets as of this year that they have put together with the Wildlife Futures program. They've also updated the herp rehab guidelines, and we're putting on this webinar series for everyone. Some of our other national task teams include the Collaborative to Combat the Illegal Trade in Turtles, as well as our Diversity, Equity, and Inclusion task team. If you're interested in finding out more information about PARC and getting our brand new newsletter, which will be coming out in August, I will drop this link in the chat. So we hope that you consider signing up and you can hear about future updates like some of these products that the Disease Task Team and our other task teams are putting out. There also is the Global Amphibian and Reptile Disease Conference coming up in Knoxville, Tennessee in August. So for those of you interested in herp health, this might be an excellent venue for you to check out. Look for advertisements too for our upcoming education campaign on Every Turtle Counts, which will be coming from our Collaborative to Combat the Illegal Trade in Turtles campaign. So with that, I am going to say welcome to our 2 speakers today. With us, we have Dr. Katie Kamen and Dr. Paula Henry of PARC's Disease Task Team. So Katie is a wildlife veterinarian with the Pennsylvania Wildlife Futures Program and the Washington Department of Fish and Wildlife. And she has specific expertise in turtle or fungal shell disease with her work with Western pond turtles in Washington. And if you had joined us with our previous webinar, you might have seen her and Dr. Dan Greer present on herpetofaunal diseases in North America. In addition, we also have Dr. Paula Henry, who's a research physiologist with the U.S. Geological Survey Eastern Ecological Science Center at Patuxent Research Refuge in Laurel, Maryland. Dr. Henry's primary area of research is ecotoxicology, including interactions between exposure to environmental contaminants and wildlife sensitivity to pathogens. So we're very, very excited to have this team here today to present to you. And in the back end, several of us will also be assisting with monitoring the chat. That includes Dr. Lisa Schender, who's a member of our PARC Disease Task Team, Michelle Christman, who is the Federal Agencies Coordinator for Partners of Amphibian and Reptile Conservation, as well as Dr. Dee Olson, another member of the Disease Task Team. So with all of that, I'm going to stop sharing and let Katie take it away. So thank you.
There we go. So do y'all see the right version, not my notes? Okay, perfect. Awesome. Thanks, Carrie, for that really awesome introduction. As Carrie mentioned, I'm a wildlife veterinarian with the Wildlife Futures Program, University of Pennsylvania. And I also do work for the Washington Department of Fish and Wildlife as the diversity non-game wildlife veterinarian as well. And I've put this presentation together in partnership with a bunch of folks, mostly that are on this call. So I do want to highlight that this is a very collaborative presentation. And so thanks to all my colleagues who have helped build this and put it together. And thanks to all of you for joining us for this very important topic. So why are we here? I think all of us on this call are very much aware that herp diseases are a priority for non-game wildlife health to protect biodiversity, ecosystem services, the whole plethora of things that herps and reptiles and amphibians provide for ecosystem services. And biosecurity and decontamination efforts really do provide us the opportunity to be proactive. In the world of wildlife health, unfortunately, once a disease ends up in a population or in an area, it's really difficult to then take that disease back out, right? Remove the disease. We end up managing for the presence of that pathogen and disease. And so prevention really is absolutely critical. And I think for herps, for turtles and amphibians, it's really, really important to be proactive, and biosecurity and decontamination give us that opportunity. And so what is biosecurity? And I think most of us here probably are quite aware of what biosecurity is. It's everything that we do to keep diseases and pathogens out of the systems. You can divide this into two different types of biosecurity. So you have your structural and your operational biosecurity. And structural biosecurity is basically, exactly what it sounds like: the gear, the nets, the boots, anything that's used that comes in contact with the animals and the environment. If you're in a captive setting, it would be the bins that they're kept in, the nets and things that you use to clean, the food dishes, the gloves that you use, things like that would be your structural. And then your operational biosecurity are the actions that you take. So what's your plan? What's your biosecurity protocol? What cleaning and disinfectants do you use? And so it's really, I think to me, it helps to separate these two aspects of biosecurity out, because biosecurity can get very complicated and can be a bit overwhelming, especially if you're thinking about doing a big operation in the field. So having these two components in mind as we go through this, hopefully will be somewhat helpful in distilling it and simplifying it a little bit. What's most important for herps and wildlife? Both aspects, both the structural and the operational, the plan, which we will talk about, are both extremely important. And then I do want to really highlight on biosecurity within captive systems. We're not covering that here. It may be a topic for a future webinar, but there are a lot of conservation efforts regarding herps deal with captive animals, right? So head starting, captive rearing for head start purposes, and then release and repatriation, confiscation of native North American species. So biosecurity within those systems is also extremely important. But we're not going to cover that here. We're really going to focus on the field aspect for this presentation. So what about decontamination? I just mentioned biosecurity and decontamination. So basically, decontamination is the process of removing the organic matter, so cleaning and decontaminating. So removing the organic matter and the microorganisms from whatever substance that might be. Your net, your boots, your hands, your gloves, your buckets, anything that you might be using in the field. So what needs to be decontaminated? And that's a question that we'll really circle back to and talk about throughout this presentation. And it really, I think, depends on the level of decontamination and the level of biosecurity that you're trying to implement for whatever the reason may be. If you know you have a pathogen present in the system and you're doing an enhanced protocol, then maybe absolutely everything needs to be decontaminated. If you are just doing the standard protocols because the risk is relatively low, then maybe just the materials that come into contact directly with the animals can be decontaminated. So keeping in mind the risk, right, and the levels that should be cleaned and decontaminated is really important. I like to even like bring up in the areas where we need enhanced biosecurity, and we know we need higher levels of biosecurity and decontamination. It's even important to wipe off your data sheets, right? So have right in the rain data sheets that you can wipe down and make sure that you remove any organic matter and potential pathogens that are present. And so the 3 levels to keep in mind for decontamination are cleaning, disinfecting, and then sterilization. And so we'll talk about those a little bit more in just a minute. Sorry, my slides are not moving. So the most important very first step that we need to do is to clean your gear. Cleaning has to be done basically so that the disinfectant that you're using can actually work. A lot of the organic material, the mud, the gunk that's on your boots or your hands or your gloves or whatever it may be, will actually deactivate a disinfectant and prevent it from working. And so what I'm showing here in this image is just a series, a study that was done with smooth sole boots versus rough sole boots. And number 1, they were not cleaned. And number 2, they were very mildly, the dirt and the mud was removed. And then number 3, they were scrubbed down and cleaned completely. And then they just took a swab and cultured everything that came off of the boots. And so what you can see is that, the dirty boots, I'm sorry, this image is kind of pixelated because it's pretty small, but what you can see is that the very dirty boots, you end up with a lot of stuff growing still, even after you've disinfected them, versus if you clean them first and then you disinfect it, your disinfectant can actually be successful in killing or killing the purpose of the disinfectant, the microorganisms that are present. I do want to highlight the difference between disinfection versus sterilization. And in general, in the field, we aren't sterilizing things. That's not really possible in a field setting. So disinfection is the removal and the reduction of microorganisms, typically focused on the ones that we know are harmful. So if we're focusing on a chytrid fungus, B-Sal or BD, then that would be the disinfection protocol would be geared towards removing and reducing the presence of those harmful microorganisms or pathogens. Sterilization is really the process of killing everything that's there, all microorganisms. And it's really very difficult to achieve true sterilization, as I have in this image here. That is the goal of what happens in an operating room, is to have things be absolutely sterile. In the field, it's not really feasible, right? We are just aiming for disinfection and a reduction of those harmful microorganisms and pathogens. So with that in mind, what really is an ideal disinfectant? I'm sure most of us are aware that there's a number of different disinfectants out there we can choose from. So how do you pick the best disinfectant for your goal, right? What you're trying to do. And so there are 4 main things that I like to talk about when I think about selecting an ideal disinfectant. And there's no perfect disinfectant. No disinfectant meets all 4 of these things at once, right? So you want something that's got a broad spectrum of proven activity, depending on your situation, right? If you're targeting, say, BD, then this may be, the broad spectrum may be slightly less important, and you can choose something that kills BD because that's your target pathogen, versus if you just want to be really careful because you don't know what's present in your system, you might go for something that's a little bit more broad spectrum. You want it to reduce the target viable organisms, right? So again, are you trying to disinfect for snake fungal disease, or are you just trying to disinfect to be extra careful because you have an SGCN species present in the system and you want to make sure you don't accidentally bring in a pathogen? And so, just thinking about what the disinfectant is labeled for and what it will actually reduce in terms of the target viable organisms. And then ideally, it will work in a wide range of conditions and environments, and also the presence of organic material. And with this in mind, it also includes contact time, right? How long does that disinfectant need to be in contact with the surface of what you're trying to disinfect to be successful? If it is going to require an hour, is that feasible given what you're trying to do? Does it remain wet? You know, a lot of times alcohol can be a really great disinfectant, but it evaporates pretty quickly, right? So can you use it and have an appropriate contact time while it still remains wet? Because remaining wet is part of, the contact time, right? Remaining active. Is it non-toxic, non-irritating? These are a couple other things I don't have listed here, but considerations when selecting your ideal disinfectant is it non-toxic or non-irritating? How would you dispose of it? And is it compatible with surfaces? Are you trying to disinfect just your boots, or are you trying to disinfect some nets, or even potentially some equipment that's really quite sensitive and or fragile? And so you want to use something that's less caustic. And I think I just went through all of these, but I'll just hit on them again really quickly. So a few more, just to keep in mind contact time. I got ahead of myself here, sorry. Contact time is really important. I know, and some of the field work that I've done and helped coordinate with wildlife biologists, contact time can be really important, right? If you're trying to hit multiple sites in a day, you don't always have the half hour to hour to wait for that contact time. And again, the non-toxic, non-irritating to the user. So this is really important, right? So we are sometimes disinfecting our hands and coming in contact with whatever the disinfectant may be. So making sure it's not, not going to be irritating your skin or respiratory tract if you breathe it in. And then persistent, right? This is something else to keep in mind. Is there a residual effect on surfaces? And if so, is that a good thing? Do you want it to have a residual effect on surfaces, or do you need it to go away so you don't, potentially contaminate an area with a disinfectant chemical? So just really briefly, I want to touch on the two main types of disinfectants. So we have physical disinfectants, which is, exactly what it sounds like. Sunlight, UV, is a really phenomenal disinfectant. Will break down the DNA and or RNA within pathogens, and so it's a really great disinfectant. Heat. The actual temperature that you need to use for heat typically depends on the pathogen that you're trying to kill. Typically, 60 degrees Celsius will work. Pasteurization, which is pressure-based and can be heat-based. Filtration can work as a form of disinfecting. If you're dealing with water, then you could potentially filtrate with a small enough pore size so that you remove the pathogen from the water. And then autoclaving can also function to be a physical disinfectant. On the chemical side, there's a handful of different types of chemicals that are considered disinfectants. So you have your aldehydes, which typically work by denaturing proteins and disrupting the nucleic acids. You have halogens, which oxidize the proteins. These would be your iodophores and your chlorines. Alcohols, they tend to denature the external membrane proteins on pathogens and then render them inactive. Your quaternary ammonium compounds, they're charged particles that chemically react and destroy other charged particles or non-charged particles. I'd have to remember my organic chemistry from way back when. And then oxidizers would release oxygen after altering protein structures. And so there's a variety of different disinfectants within these chemical classes. And I just want to highlight, that the use of chemical disinfectants in aquatic settings must adhere to federal and state laws and regulations that govern the sale, use, and disposal of those disinfectants and stimulants. And that brings us to the importance of labels. And so most all of the disinfectants that we've mentioned and that we'll talk about have a label that has been approved by the EPA. And so with it, it's very similar to the drug approval process. So if you take an approved drug, it comes, with that product information that has all these warning labels and information from trials that have been done to prove, scientific data to show evidence for the label claims. And so these disinfectants are pretty much the same, right? So they have specific claims that relate to the effectiveness associated with that claim. And often contact times would be present on the label itself. And then they would have to list the active and the inert ingredients. And sometimes these are proprietary, so it's not completely linked if they're under, whatever the proprietary protections are for that. Then these precautionary statements, often based on like the MSR, no, I can't, whatever the document is that outlines, various precautions in human health and animal health risks. First aid information. So if swallowed or if contact with skin, this is what you should do. EPA registration number, which you can then Google and go to the EPA and get typically all of the information and the studies that have gone into making this label are available. And then directions for use and often disposal. And disposal, as we all know, is frequently really quite difficult because often on the label, it just says dispose of in accordance with your local rules and regulations or laws, which is often not very helpful. But it should have, these labels should have some disposal information as well. And again, just to highlight, because I think we have to, when given this type of a talk, it is a violation of federal law to use a product in a manner that is inconsistent with its labeling. So just keep that in mind. So gear disinfection, just to kind of recap a little bit about what we've been talking about. So if you have, you've been in the field and you're now back at your site, the site that you set up for dealing with biosecurity and cleaning and disinfecting, when you get back, the very first thing to do is to clean. So just water, brush, scrub brush, try and get all the organic material off of, say here, I've got some boots shown. And depending on the level of the biosecurity and the decontamination that you're dealing with, you may have a tote simply for washing, right? So that you capture all of that organic material that's coming off of those boots or the gear, whatever it may be, because you may want to take that back and disinfect the water itself, right? If you're dealing in a high risk area where you have a known pathogen of concern. And then again, just thinking about how you select your disinfectant. I think we've hit on this in previous slides, but things to keep in mind is contact time. Is it feasible? Is it going to damage your gear? Something like bleach is really, really great, but if you're disinfecting nets repeatedly, then it might wear down some of your gear. So just keeping that in mind. And then how often you need to remake your disinfectant, right? On a bleach, it's remake it every day versus something like Virkon X or Virkon Aquatic, you could potentially it might last a little bit longer, but it's really important that you clean it first because that organic material will lead to the degradation of your disinfectant. I just wanted to highlight this, and many of us on the call may be familiar with this publication from Gray et al. 2017. And I think Carrie or someone will probably stick this in the chat because it is available on PARC's website. But there's this really awesome table. Sorry, I don't know if in your view this is in your way, but I'll move it. But there's this really awesome table that several pages of an appendix 1 that basically goes through all of the disinfectants that are currently available for use on herps and the concentration, the contact time that would be needed, the application. So here, you can see that Virkon Aquatic is really safe for field equipment. The efficacy, move this, would list, which target pathogens this for this little snippet that I have. It's Virkon, ranavirus, BD, snake fungal disease. It's efficient or efficacious against those 3 pathogens. And then these, the links, or not links, sorry here, but the those numbers are, brain is escaping me, words are not coming to me. But these numbers are basically the references, right, for the studies that showed that Virkon will kill ranavirus in this contact time. And then it has human health, environmental risks, and what you could do potentially to mitigate those negative effects. And this is an extremely useful table if you are, putting together your biosecurity and decontamination protocols, and you're like, what disinfectant should I use? This really is extremely useful. So I highly recommend snagging a copy and just having it on hand. I do want to highlight here about bleach. It's really important if you're going to be using bleach as a disinfectant that you are diluting at 1 part of household bleach. So just regular old household bleach. Make sure it says household bleach on the container. Bleach now comes in a variety of different flavors. And so you want to make sure you get household bleach, and it's 1 part of bleach to 9 parts of water. And you also want to make sure that when you buy that gallon of bleach that you're replacing it pretty, periodically. I know that we say to remix this 10% bleach or 1 to 9 parts bleach on a regular basis every 24 hours. But the jug itself of the, concentrated household bleach or standard household bleach, is only good for, about probably a month or 6 weeks or so, because it does also degrade over time. And so just making sure that the containers of bleach you have are not too old. So in general, and I think we all know this, but to remind us all, why is decontamination and why are these biosecurity protocols so important? Really because, if we're moving animals about, if we're moving ourselves about, then we could potentially be moving pathogens around, right? Both those that we know exist, but also those that we don't know. And I think that the unknown is every bit as important as the known, right? If we're going out in the field and, we're in a site where we don't know what may or may not be present, but we have, like I mentioned before, an SGCN species, then I think it's really critical to consider, an enhanced biosecurity protocol to protect, the potential movement of a pathogen into that system that might impact that SGCN species. So when to decontaminate, and this is something that we'll touch on in several different ways throughout the rest of this presentation, but how to define a site. So I think, when you're thinking about creating your decontamination and biosecurity protocol, one of the most important things to consider right out of the gate is how you define your site. And that's going to be based on host and pathogen ecology, dispersal distances, water movement, all of these different things that play into how you might define the site. And it really is important to consider, again, your host and your pathogen ecology, right? Because if you're working in an area where there's numerous ephemeral ponds super duper close together, it may not make much sense to do enhanced biosecurity between those tightly linked ponds or sites, right? Because the animals are moving between that anyways, versus if you have an isolated stream or like an isolated area that may be, upriver, and, some pristine upriver watershed areas, then it might be very worthwhile doing enhanced biosecurity in those areas. Thinking about aquatic considerations, what are the water flow conditions? What's the human use? Are there a bunch of people with their dogs hiking in and out all the time? What's the overlap with the terrestrial system? And the same is true with terrestrial conditions, right? What's the habitat use and connectivity? What's the human use? How does that overlap with potential aquatic sites nearby? And so I think it's, just these things are what really do make establishing a biosecurity and decontamination protocol difficult, but they're absolutely critical, right? To make sure that the protocols that we're putting in place are effective and meeting the needs that we want them to meet. And knowing the risks, right? I think that's also really important. Do we know what pathogens we're concerned about in these areas? Do we have susceptible hosts? Because there are situations where the pathogen might be present, but there's not a susceptible host. So in that situation, your protocols may be a little bit different, right? Because you're really at that point trying to contain the pathogen to that site, but there are no susceptible hosts. And then if there's a history of disease, do we know, say, BD has been present and has caused a mortality event in this particular population? If so, even if we haven't detected BD in a number of years, it might be worth being very careful when you go into that site because it does have a history of that pathogen. And then I tend to be very focused on pathogens as a wildlife veterinarian, but I do really want to highlight that pretty much everything we're talking about also applies to aquatic invasive species and invasive species in general, and hitchhikers and things like that. So all of these decontamination biosecurity protocols could be for pathogens and preventing disease. They could also be and should be implemented for aquatic invasive species, even though I'm kind of blinded because I'm a wildlife vet. And so with that, I can take a brief break to see if there's any questions before we launch into the last few slides.
Yeah, Katie, I do have a couple of questions that I've captured from the chat, so I'll kind of group those. We have a couple of questions that pertain to bleach. One person said that they like to make sure that they buy a disinfecting bleach, not the kind that's intended for use of laundry. So I guess that's more of a comment, unless you want to, provide any additional information on that subject.
Yeah, no, I think that's a really good point. And I think just being careful for the type of bleach that you purchase. And if you look at the Gray et al. Paper, that appendix that I mentioned, when the row for bleach, it goes into, or it highlights some of the differences in the different types of bleach. But there are bleach products out there that actually don't contain bleach. So I think just being really aware of what product you're buying and making sure that it's the concentration of the standard household bleach product. So then when you mix your 1:9 dilution, you're getting the 10% bleach solution that you need.
Right. And then as a follow-up, someone else said that they're concerned that using a Clorox mix could damage their waders, especially if it's for use over a whole field season. Do you have any experience with that, or recommendations to reduce degradation to waders?
Yeah, that's a great question and a really good point. So bleach is very caustic and it will damage gear, especially if it's used repeatedly over time. So if you have sensitive gear, there are other products that can be used. Virkon Aquatic is a product that's pretty safe for gear in general. So it may be that, if you can divide the disinfectants, right? So you can pick what if you need bleach to hit nets and things that may be more critical or whatever. Like if you want to divide it, right? If you want to use just bleach, because Virkon Aquatic is also expensive, but there are options that are less caustic, right? And the other benefit of Virkon Aquatic is that once it's mixed, it does last a little bit longer than the 10% bleach solution. But yes, I think that if you're dealing with gear that you don't want to degrade a lot over time, trying to use something other than bleach would be beneficial.
Right. And then moving on to the use of alcohol as a disinfectant, Mike asked, what are your thoughts on using isopropyl alcohol as opposed to ethanol? There's apparently more literature on the use of ethanol, but less available information on isopropyl.
That's a good question. And I would have to refer to that Gray et al. Paper with the review of the different disinfectants for isopropyl versus ethanol. But I would try and focus when you're selecting using isopropyl or ethanol, using what's in the literature and has, proven contact times and things like that. That doesn't really answer your question. I'm sorry.
Okay.
I don't know if any of the other folks like Dee Dee or Dan or anyone else on the call or on with PARC has input or thoughts on that question more than I do.
And if not, we'll probably be compiling some of these questions and we can follow up with things that we didn't know immediately off the tops of our head during this Q&A period. So let's see.
Lisa, Patrice Klein mentions that alcohols are not effective against spores. Hydrogen peroxides are effective against spores and can be more efficacious disinfectant.
Okay. Thanks for, I'm looking at a different document, so I'm missing what's coming in on the chat. So thanks for that. And then we also have a question about advice that pertains especially to disinfecting sensitive items like dissolved oxygen meters, pH probes, and whether we might consider using like UV sunlight for those devices.
Yeah, that's a very good question. For those devices, I probably would use something like UV. I would combine it with like an alcohol disinfectant wipe that, hopefully would not scratch or damage the probes. And then, obviously cleaning them with fresh water, and then, heat or disinfectant, or I'm sorry, UV for the actual disinfectant, I think would be the better approach than, subjecting them to something like bleach or even Virkon, since we don't know the impacts of those chemicals on the probes.
Right. And then there was a question earlier that's kind of maybe open for broader discussion. So feel free to table this towards the end of your talk, but for people to be thinking about it, the information that we're discussing, is it important to share this with other survey teams like wildlife and people that are out doing vegetation so that those folks would know this disinfection protocols, even though it might not pertain especially or particularly to herps. And that's something that we've discussed in other meetings. So yeah, big topic there.
Yeah, that's a great topic. And yes, that would be my answer is yes. I think it would be great to get, other folks that may be outside of the herp world, but still do work in the field. I think a great example that always comes up is, there's, I don't remember who relayed the story, so I'm sorry if I'm stealing your story, but the moose biologist who's like, oh, I don't have to worry about herp diseases because I'm just dealing with moose. But you're like, you're trapped, you're going out and walking through all these aquatic environments that are very herp specific and very critical for herps. And the same is true with like beaver, right? So I do think that getting this out more broadly for folks who are just generally in the field, right, and may have overlap or do have overlap with critical habitat and important habitat for herps would be really good.
Yeah. Carrie, do we have time for a couple more questions or should we hold off for now and move on?
I'd say maybe one or two more.
Okay, so the next question is asking about a product called Star San, which I'm personally not familiar with. And the question says that this can be used as an alternative to bleach when cleaning beer making equipment because it doesn't damage components and kegs.
Huh. I've never heard of it.
Okay. Well, maybe
so
Robert, maybe you can post a link in the chat for those of us who aren't familiar with this so that we can look into that later. And then one final question for now from Madeline. I always hear different advice for the shelf life of mixed Virkon. What do you recommend as the maximum amount of time post-mix that Virkon will remain effective?
That is a very good question. I tend to say, I guess there's two things I take into consideration. One is how often it's being used. So if it's a foot bath and boots aren't being cleaned between, you're like stepping in it and then walking on, then I think it should be changed every 24 to 48 hours just because of the organic materials that are in it. But typically I would say a week or so is sufficient, if not a little bit longer. I know that at least in the past, there have been test strips that were available to show the activity of the whatever the chemical is in Virkon Aquatic. And so if those are still available, then that would be another option, is to get some test strips so that you can test and make sure that it's still active before you replace it. But in general, at some of the facilities I've worked with, it's just kind of standard practice to just replace it once a week. If it's, where you're dipping your nets or dipping your waders or just general cleaning, then that tends to be replaced on a weekly basis.
Okay, and I see that Clinton has shared a link to the Northeast PARC website, which also has some disinfection protocols with some shelf life information. So that might be a place to look as well. So, all right, let's move on with your presentation now.
Okay, so now we can launch into the plan, which is, it's complicated. The plan is always complicated, I think. So I think, what the importance of the plan, and that's, to me, in my mind, it's absolutely critical to have a plan, because if you have a plan, then as you're going into the field or as you're doing whatever the work that may be that you're doing for that requires disinfectant and biosecurity, if you have your biosecurity plan in place, then you can be organized and you can have everything set up and you have your disinfectant already selected and how you're going to do it, and the various containers, like I showed in one of the previous slides, the container, the tote for collecting the wash water from your boots, and the tote for collecting the disinfectant from your boots. Really helps to have it, to help things move pretty smoothly when you're in the field and already really busy and probably tired because it's been a long day. And it helps you to, you know, identify the risks, like I mentioned in the first part of the presentation, right? Do you know what pathogen you're concerned about? Do you know what species or host species you're concerned about, and the ones that you're trying to protect, excuse me. And so I think, you know, just kind of walking through the plan and having it be in place and made in the office before you ever even get into the field is, I think, really important, because it will help you identify where there may be gaps. And I, you know, I think it's also really important to keep in mind that, you know, you may sit down in the office and spend a couple of days building your plan, and then go into the field and be like, well, that didn't work. And so you have to come back and replan and just redo it. And that's perfectly normal. And I think, you know, how we get to having optimal biosecurity plans in place, right, is, you know, trial and error. This flow diagram is also from the Gray et al. Paper, and it outlines kind of the very basic steps that you should consider when you're starting to establish your biosecurity plan. So you start out with, do you know you have a known contaminated site? No. Are there species or taxa with potential known or unknown risks? No. Okay, then maybe you can just do, you know, the minimum, very standard disinfectant and decontamination protocols between sites, kind of as the minimum, right? But, you know, these yellow boxes, and I'm not going to go through all of this, but the yellow boxes will lead you to this purple box, which really is what we're calling the enhanced or elevated protocols, right? So the strictest of the protocols, where you're going to be changing your gloves and decontaminating equipment between sites and wearing PPE, including even like potentially a full-on Tyvek, changing or using different gear between individual animals, and then not moving between sites until a full decontamination is done, including of your, you know, your trucks and your potentially bigger equipment. So I think this is, you know, it's pretty simplified, but I do think that this is a kind of helpful flow chart when you start thinking about, you know, what you need to incorporate and what may guide the risk level, right? And the level of biosecurity or the protocol that you put in place, enhanced versus standard versus somewhere in the middle. And so some more considerations for this plan level, and this is from a Olson et al. 2021 paper that really looks at elevated or enhanced biosecurity practices. And so, you know, if you're looking at pathogen export, so do you have a site where you know you have the pathogen of concern present, and are you worried about taking that pathogen of concern somewhere else? If yes, and you're going to pathogen-free sites that are high risk, then maybe you need to have this enhanced biosecurity protocol, versus the pathogen is present, but, you know, the next site you're going to doesn't really have high risk taxa, maybe the host species aren't even present, then you can just, you know, implement some standard biosecurity practices to reduce the spread, the potential spread, right? And the same is true kind of on the reverse side if you're looking at pathogen import. So if you're, you know, at a site where the pathogen is present at the site, no, but you have host species like STCN species that may be high risk, then you want to use your enhanced biosecurity practices. And so this is just another kind of simple, you know, risk assessment flow diagram that can help you really think about what level of biosecurity you should be putting in place for your plan. And some considerations, I'm sorry, this is an extremely wordy slide, but just some considerations to keep in mind. And I think we've covered a lot of these. So I apologize for repetition, but, you know, repetition is the key to success, and it's how my brain remembers things. So basically looking at, you know, the pathogen and host surveillance, if you can, right? If you're in a study area, do you know what hosts are there that might be susceptible to your pathogen of concern? Do you know if the pathogen of concern is there? If you don't know, but you have a high risk or, you know, an SGCN species, then, you know, you want to consider enhanced biosecurity practices and starting with the lower risk sites first, right? You always want to move low risk to high risk so that you're not potentially spreading things around in terms of the pathogen presence, right? So if you know you have a pathogen present at one site, then start at sites, you know, that may be unknown and move to the pathogen present site last. It's kind of reversed if you have SGCN or highly, potentially highly susceptible taxa that you're trying to protect. Then you might want to start with them first, right? If it's, it's like an SGCN species that is highly susceptible to BD, you might want to start with that site so that you reduce the risk of bringing BD into the site. And again, how you define your site is going to be based on the epidemiology of the pathogen and the host species. And you always want to decontaminate between sites. Once you've defined what your site is, then you always want to do as enhanced as possible decontamination and biosecurity between your sites. And then, there are biotic considerations and abiotic considerations to keep in mind. Biotic being, pathogens present, host species present, these things that we've been talking about. The abiotic considerations kind of would be your structural, what I mentioned previously is like structural biosecurity considerations. So your gear, your nets, your data sheets, your hands, your vehicles, things like that, that, are the abiotic structural components. And then think about, the necessary level of decontamination. Are you so concerned that you need to do decontamination between every animal? Or, if you're working in a site or a series of sites where the pathogen is present, but the host species is not highly susceptible to it, then maybe you don't need to decontaminate between each animal. So these are just kind of the different things, to keep in mind as you're actually putting your, pen on paper and writing down your plan. And this, again, it's where it gets quite complicated, and in some ways can be a bit overwhelming. Sorry, I skipped a slide. Go back. And so I think this is from this is a snippet from table 1 from that Olson et al. 2021 paper that I mentioned, looking at field site contexts for developing your risk assessment and your level of biosecurity required. And I think this is a really it's a really fantastic table that goes through 7 different scenarios, and based on the outcome of the scenario, whether or not you should do enhanced biosecurity or just kind of what we call the standard normal biosecurity. And so I'm not going to run through all of these just for sake of time. And I think you can access the paper. It's available on the PARC website. But for example, with this pathogen occurrence, is there a disease die-off at a known site? If yes, then that might warrant enhanced biosecurity to reduce the spread of that disease or pathogen-associated die-off. And then, pathogen habitat. Is the pathogen present or not? If it's not present, then maybe, a low priority and kind of, a lower level of biosecurity would be okay, unless there are other contexts that play in, like, is there an extremely rare host species in that habitat that you really need to protect? And so I think, take a look at this table. I think it will provide some pretty good examples and some, good guidance for helping develop your risk assessment when you're thinking about the level of biosecurity you might need to implement. Okay, and then so what is an elevated plan when you're thinking about having an enhanced biosecurity plan? And this is just kind of the elements to consider when you're dealing with this elevated plan. So moving low risk to high risk, right? So if you are working in a system and you have a site that's a very low risk site for a potential pathogen, you're going to want to start at that lower risk site, host species that are lower risk. You want to start at that lower risk site, right? So that you are able to implement the strict biosecurity practices as you move to a higher risk site where that potential pathogen may be present, right? And that really helps reduce the risk of moving that pathogen that's present at the high risk site to the low risk site that we know the pathogen isn't present. Hopefully I said that correctly. I think I got a bit tongue-tied there. Housing individuals separately. So if you're doing capture efforts and you're in a really high risk area where there's a pathogen of concern present that has the potential to kill the hosts that you're working with, then you're going to want to keep those individuals separate, either in separate bags, snake bags, or the Ziplocs for amphibians, or buckets if you're dealing with turtles. So house them separately as much as possible and disinfect or don't reuse the containers that you're housing them in. Wear gloves, change your gloves between individuals, wear a Tyvek that you can take off and dispose of in an appropriate manner before going to the next site. Any other PPE that might be necessary. Maybe don't wear the same boots between these higher risk sites, right? So have dedicated site-specific boots. If you really wanted to go all out, you could have dedicated site-specific gear for sites that you know you have the pathogen of concern. I know here in Washington, that's what we've done with white nose syndrome in the counties where we have white nose syndrome. And sorry, it's not a HERP pathogen, but if we know we have white nose syndrome or PD in those counties, the gear we use for handling and capturing bats don't leave those counties, or even within the county, they don't leave smaller areas. And then store your trash and dirty gear for proper disposal. You want to double bag it. You can do your first bag, disinfect that. Second bag, disinfect it, right? So having your wipes on hand for disinfecting the outside of the trash bags. And then consider, the water in the soil is also a risk, right? So if you've just washed off your boots and your waders into the tote, you now need to consider how to disinfect that water, that wash water from the tote. So can you use Virkon? Can you add enough bleach to get it to the appropriate concentration to make sure that any pathogens that are in that water are then killed so that you're not spreading them? And then I think, there's other things to consider, I think, that I have not covered here. So just keeping in mind that, the plan is always something to go back and revisit. And another, just to highlight this Olson et al. Paper again, there's a second table in this paper that really outlines the differences between what we would consider standard versus enhanced biosecurity procedures and protocol or plan. And I think it is a really great kind of stepwise table or process outlined in the table that includes planning at the office and how you would stage at a vehicle site, what you would do upon return to the vehicle, and then again reworking the plan once you're back in the office and you're like, that did not work very well, let's revisit that. And so I would suggest taking a look at this paper, because it does really provide some great at least scaffolding for building your plan out if you're looking at standard versus more enhanced procedures. And I had to throw this slide in too for what to pack for the field, even if you're not doing HERP work. I think what we've been talking about up until this point has really been HERP focused, right? So if you're going in the field and you're doing HERP related specific work, then you're going to have a lot more in your backpack than this. But if you're just going out into the field in general, and you're not necessarily planning to do HERP related work, and you don't have like a full-on disinfectant biosecurity plan in place, you're just going out for some observation type work or something, then I think you should still have a field kit for biosecurity and disinfection work, right? And even for collecting samples in case you come across sick or dead amphibians or reptiles. And so I think, this should all fit within probably a gallon-sized Ziploc. So you don't need to have a huge amount of stuff, right? A couple of Falcon tubes, I think, would be pretty helpful, potentially for putting used gloves in, if you do use your gloves, or samples if you are to collect, if you find some dead tadpoles, it gives you something to put those in. These scrubbing bubbles can be really useful. They are not, I don't believe they are not in the Gray et al. Paper, but they can be really useful because they do kind of clean and disinfect at the same time, just given the mechanism of action, how they work. And so if, in a pinch, you really need to clean your boots, you can have this brush, small little hand brush. You can brush off as much of the debris as possible and then use these scrubbing bubbles. You can also have disinfecting wipes. I think this is one of those products that says Clorox, but it's not actually bleach. And then, have your gloves. And it should all pretty easily fit in one large Ziploc. And then you can also, if you think you might come across samples that you would want to collect, you can get those ice packs that you break and they can turn cold, so that you could cool them down quickly to preserve them for submitting to the lab. We are planning to build a fact sheet for this in general, just like a what to pack for the field for field biosecurity work. like the kit to take into the field. So hopefully in the next couple of months, that'll be something, a resource that will be available on the PARC website. So just to really kind of briefly recap, because this is ended up being a longer presentation. Obviously, biosecurity and decontamination are very important. There's a lot of us here on this call that are very much aware of just how important those two things are. And it is really the chance that it gives us the chance to be proactive, right? And to take proactive steps to reduce the potential risk and threat of moving pathogens around. I think it's really important to know the level of your plan. So are you just doing standard? Are you doing enhanced? Are you doing somewhere in the middle? When in doubt, I think it's best to go as enhanced as possible. So, more is better. More is always better. So when in doubt, go with more. But I do really want to highlight that anything is better than nothing, right? So if you're too busy, you're too all out, you can't do anything but have that super simple kit in your backpack to, brush off and spray down your boots with some scrubby bubbles, that's better than nothing. And so I think just remembering that anything is better than nothing is absolutely critical. And I think with that, one last slide. And I do want to highlight, this is a pretty cool picture that Dan shared with me from Megan at USGS of potential botfly infection. But it's really important to keep in mind biosecurity and how important biosecurity is in the face of unknowns, right? If we are already implementing pretty enhanced biosecurity practices, then we're very much reducing the risk of us spreading around pathogens that we don't even know anything about. And I will put a plug in here as well. So if you come across any of these unknowns or sick and dead herps in your local jurisdiction, then you can use the herp disease alert system to report that, and PARC disease task team will help connect you back with the local jurisdiction government entity that can help you collect and potentially process those samples. And I think with that, I will be done and take any questions.
All right, we're running a little bit behind, so I'm just going to ask a couple of questions that pertain more directly to what you've discussed, and then we'll hold off on some of the broader questions till after our second speaker has gone through her material. So the first more pertinent question to the topic was in regards to disinfecting nets, and if you have particular products that you recommend for disinfecting nets between captures. And maybe how long you think that it needs to, sit on those nets for disinfection to occur.
Yeah, that's a really good question. I think it would depend on the type of net. So, obviously, if a little hand net, you can just have a little tub of, Virkon aquatic, a chlorhexidine or something like that you can just put that type of equipment in. If it's something bigger, like a fike net or something that you're using for the actual capture itself, I personally really like Virkon aquatic. I think it's less caustic than some of the other disinfectants out there. And so I would, you could have one of those little spray things that people will use for like spraying fertilizer or chemical pesticides, and just really wash it off, make sure it's clean, and then really soak it down really well with the Virkon aquatic, give it the 20 to 30 minute contact time, and then wash it off with fresh water and allow it to sit in UV for a period of time, a couple of days if possible. Then I think that could be a pretty solid disinfection protocol before moving between sites. Again, if you have if you're using these nets and really known high risk sites where you have really wicked pathogen present, like ranavirus, say, I would, if it's at all possible, I would recommend having those nets be dedicated to those high risk sites, and then have other equipment that you would use for other places that may not be high risk given the pathogen presence.
Great. And then before we move on, I just want to highlight a couple of questions for you to be thinking about for the end of our discussion, not necessarily to answer now. So one question is in regards to working in urban parks where there are high levels of human visitation, and anything that's known, case studies, et cetera, from herp pathogen presence or speed of dispersal in these sites. And Dee Dee's already answered some of this in the chat. So we'll move on for now, but just please be thinking about that. And then a second question from Emily is about wildfire or wildland fire aquatic invasive species prevention standard operating procedures, and whether there are gaps or areas of improvement that might be feasible to implement for biosecurity purposes. So let's hold off on those two questions for now so we can move on to the next speaker, but hopefully we'll have time to come back to these and have a more discussion about that in a little bit. So yeah. Thanks.
Great questions.
Thank you, Katie. And we're going to hear from Dr. Paula Henry, a research physiologist with the USGS Eastern Ecological Science Center at the Patuxent Research Refuge in Laurel, Maryland. And Paula, we see your screen. Can you please hit the slide share and take it away?
Yeah, hold on a second. I can't do it. I'm sorry, I'm having problems here. Okay. Shoot. Okay, well, it disappeared now. You can't see it, can you?
That's yes. That is the public urban parks public visitation question is a very good question, right? Because it's one of those things of like, well, if people are coming and their dogs are going in and out of these parks and waterways, then what good does biosecurity do? But I think in those situations, it's also about educating the general public. So if you're out there on the ground, working in these situations and somebody sees you washing your boots or gaiters off or whatever, then that gives you the opportunity to talk to them about how important biosecurity is, and maybe they should think about washing their dogs when they get home, or not letting their dogs swim in those environments.
Yeah.
As far as the spread and the dispersal and speed of dispersal in these types of sites, I would have to rely on Dee or Dan for that, because I actually don't have that information on the top of my head. It looks like Paula's presentation's working. So I'm not sure that fully answers the question. I think we can continue discussing it after Paula's presentation.
Okay.
Looks great, Paula.
All right. You're in the middle, but that's okay. Okay, so I'm Paula Henry. I work for the USGS Eastern Ecological Science Center at Patuxent in Laurel, Maryland. And Katie's presentation was great because it certainly introduced my presentation in terms of many of the steps that we're going to take. So, why isn't it working? I cannot move this forward. Why is this not working? Okay. So I'm going to give a brief review of why biosecurity is important when working with heavy equipment, present the when, where, and how to decontaminate, and also how and where to plan ahead to minimize the spread of diseases. And planning is, of course, a big part of this whole presentation today. So biosecurity in the context of large equipment refers to activities such as habitat, dam removal, road construction, fighting wildfires, installation of wind turbines or power grids. This means that there is a shift from cleaning and disinfecting field supplies, such as your boots, to manipulating heavy equipment. This increase in scale will mean that you need to plan where to set up, where to use and relocate the equipment over multiple work sites. All of this without the unintentional consequence of contaminating a clean wetland or aquatic habitat. So I think I skipped a slide. Yes, there are many herp pathogens. So I'll just list briefly, mention 3 groups of pathogens that have been and continue to be of high concern, and most people will know what they are. The chytrids, such as BD and B-Sal, microscopic waterborne fungi that have been linked to amphibian species decline and extinctions. B-Sal is thankfully not yet in the United States. Ranavirus of the iridovirus family is a large family with at least 3 amphibian-specific viruses, including the frog viral 3, and it affects fishes, amphibians, and reptiles, and ranavirus can persist outside the host, as well as severe Purkinje infection, SPI, the 3rd most common infectious disease of amphibians, and it infects tadpole stages only. And without going further into the organisms themselves, although biosecurity details may vary as we learn about newer pathogens, emerging pathogens, the general concept for biosecurity and the recommendations for different steps will pretty much remain the same. In other words, it's pretty important. So how are we as humans contributing to transmission? Certainly Katie covered some of that, fishing tackle such as crayfish, the pet trade or dogs walking in a wetland or in an urban setting, commercial, farming operations, field research like surveillance or monitoring activities of even birds, any other wildlife like moose maybe. Fomite control is when local water is used for wildlife activity, fighting wildlife, or even for controlling road dust. Translocation of amphibians and reptiles, which is really a fancy way of just explaining picking up a turtle at outside my house and moving it to my work site. But the focus today is primarily that I am presenting is primarily, on traffic from larger, mainly construction equipment and machinery that unknowingly can transmit or introduce pathogens to remote or pristine sites. The reasons for biosecurity with heavy equipment, again, overall costs. It's much less expensive and less time consuming to prevent the introduction of pathogens into a habitat than it is to eradicate or introduce pathogens, if that's even possible. Unpredictability, the pathogenicity or potential ability to infect can range from being 0 to being catastrophic to a community of wildlife, and it depends on the host and virulence of that pathogens. The host species may have different levels of sensitivities depending on their stage of development or even on their history of their previous exposures to alternative stressors. The virulence or seriousness of the infectious disease, the infection or disease, depends on environmental conditions like precipitation, temperature, soil, and aquatic characteristics. As a pathogen such as virus spreads from one host to the next, it can mutate into different variants, as we have all learned in the last 3 years with COVID. The third is containment. Like forest fires, if present, you really want to be able to contain any spread of the pathogen, and to do that effectively, you have to be prepared before it spreads uncontrollably. So in remediating an eroded stream bank, for example, you don't want to create another source of damage. Pathogens are opportunistic, so if we allow them to spread from one location to another, they will find a host. So what does this all mean in terms of locating and moving your heavy equipment? Well, here is an aerial photo of several different types of waterways that may be on a refuge or public land. Different construction activities may be taking place at site A, B, or C. And at some point, site A is done with using its ditch digger. That is now needed at site B. The contractor should consider not only what type of habitats are between A and B, but also pathogens that may be present along the way. This includes not only trucks, but also construction mats or platforms set down to minimize disturbance while driving equipment across the habitat or the vegetation, for instance. Using and moving equipment with the risk of introducing pathogens to a naive site, that is where we want to focus to cut the spread of transmission when we're talking about these activities. The contractor will need to plan ahead for when and what type of equipment is needed. So again, it is critical to plan ahead. This is a decision tree, like a little more complicated than Katie's, but in the boxes, they have the questions on what the current site is like, the presence of the pathogens, the waterways present, and where the next site is located. The blue arrows are yes answers, while the gray ones are no. Responding to the questions will help evaluate the level of risk of transmission, and I'll explain how. On the one hand, if there is a known pathogen where you are working at site A, and site B is remote, unconnected, pristine site, then the risk of transmission or introducing the pathogen to the new site is high. Alternatively, if the site A has absolutely no water, no aquatic pathogens, no pathogen history of any diseases, and you're going to move directly to a very remote site, the risk of picking up and transmitting a pathogen to that next site is fairly low. And all the potential scenarios in between fall under a significant risk of introducing the pathogen. This decision method obviously is not foolproof, but it is a good start for planning which biosecurity steps to take. So what does all that mean? Well, low risk of transmission means brush and clean, rinse and dry, and then you're done. However, if you have significant risk, you may need to add a disinfectant as well. And the amount of rinsing needed may increase, and all of that increase the cost. In fact, if there is a high risk situation for which you know the disinfectant will be needed, it may be more cost effective to rent a second ditch digger. For instance, one of the questions was, if you're working all day, you might want to take multiple sets of boots or multiple nets so that you're going from a low contaminated to a high contaminated. You don't always have to wait until it's disinfected and dried. But going back to this, what do these steps mean? Well, step one is cleaning. And as with the boots and other field gear, you need to remove as much of the mud from the vehicles so that you can expose the metal. For heavy equipment, that might mean that you need to use shovels, pry bars, or even high pressure air spades. You also want to detach the accessories such as track pads off the bulldozer, for instance, before you begin to clean from top to bottom and from the outside of the vehicle to the inside. So why plan ahead where to clean? Well, there's several reasons. First of all, you want to find a convenient location where you can sort of have supplies ready to go. And it has to be big enough so that you can maneuver these large, heavy vehicles. And finally, you want to pick a site in which there is very little risk of having your particles that take your mud going back into a stream, or the chemical disinfectants wash into a naive stream or aquatic habitat nearby, because some of these chemicals are very toxic to wildlife, including invertebrates. Step two is disinfecting. Then again, the chemical is to reduce infectious microorganisms that may be present. Whether it's needed or not, again, you can refer back to the decision tree, or if you're unsure, add the disinfectant, in fact, that gold standard. Since these are chemicals, remember to learn the hazards associated with handling, spreading the disinfectant, and storing the chemicals. All that information is on your safety data sheets that come with the chemicals. Step three is rinsing, and it is extremely critical for extending the life of your equipment, your vehicle. And certainly don't use the water from the site in which you were just working. Step four is drying. Now, Katie mentioned some ways to disinfect and dry, such as autoclaving. Well, these are absolutely not feasible for these large equipment, but artificial heating is also very expensive. So the best is outdoors in direct sunlight. And the rule of thumb is a minimum 5 days, if you're not using any disinfectant, 24 hours if you are using disinfectant. Now, again, we're talking large vehicles, so the cost of the disinfection may be less than the daily cost of actually having your equipment idle, your work stopped. So that part is also part of the planning that the contractor or resource manager needs to think of. In summary then, to minimize or reduce the spread of pathogens when using heavy equipment, first of all, you want to check the overall site topography, what type of watershed it is, how many and where the waterways are. You want to obtain data on potential pathogens present. This can be just information in the literature, or if needed, you might even want to do a eDNA or use a tool like eDNA to figure out whether pathogens are present now. You want to gather information on the community of amphibians or reptiles or anything that's present at that site. So to determine whether it's the right, whether they're at a particularly sensitive time stage of development, such as a tadpole stage, whether it's spring or fall, and when are they the most sensitive and if they'd be exposed. You also want to obviously type of equipment needed, select that depending on the scale of your job. And finally, decontaminate. And again, what are the steps to decontamination? Well, they are brush and clean, disinfect, rinse a lot, and then dry. Now, whether you're a resource manager contracting for a restoration project or a contractor planning a job, ideally, the biosecurity should be discussed during the pre-construction meetings, part of the permitting process or the work contract itself, or even better, as part of the bidding proposal. And this is obviously somewhat new, and we kind of would like to start looking more into having this as part of the bidding proposal. And I can't remember the words, but so that your bid has that extra in your contract. But for all of this to work smoothly, again, the take-home message, as before, is to plan ahead. Questions? I would like to actually recognize and acknowledge Dr. James Julian, who is present today. He's at the Pennsylvania Department of Conservation and Natural Resources, as well as the rest of the authors of the manuscript that is on the publication that is on the large equipment disinfection. And he is actually here to answer any questions that I won't be able to. Thank you, Jim.
We haven't had any specific questions pop through quite yet on disinfecting large equipment. Let's see, we'll give a few minutes here, but I'll go back to one of the earlier questions that we did have. Patty asked about, similar to the white nose syndrome decontamination protocol, can hot water be used for disinfection after cleaning off gross organic matter? And Dee Dee did reply with some information from a paper from Gray et al. About heat being useful for BD and ranavirus, with some specifics in there. Dee Dee, was there anything that you wanted to add to that, or Paula or Katie to further discuss that question?
In the Gray et al. Paper, it said the hot wash should use 60 degrees C water or greater for 30 minutes. And I think I put the greater equal sign for the time, and it was actually for the temperature. So I'm sorry about that.
Okay. Thanks for clarifying. Let's see if we've got anything else. I guess we can go back quickly to that question about standard operating procedures and gaps or areas of improvement that would be feasible to implement with respect to wildland fire, aquatic invasive species protocols. Kind of a big question there, open for discussion.
This is Katie. I was hoping that Paula or Dee Dee or some folks that have been involved in some of these discussions with the wildfire questions would address that, because that is a really good question. But I think there are other people on this panel that would be better to answer it than me.
Well, this is Dee Dee. The one activity that is foremost in my mind is when they're moving water around, and some general guidance that we have put out there is if they're moving water between watersheds, and if we have no information about the occurrence of pathogens in the destination watershed, then they should consider disinfecting the water. And that can apply for pathogens, disease-causing pathogens, but it really helps with some of the aquatic invasive species as well. So I think there's a practicality clause when you're thinking of disinfecting large quantities of water draws from nature. And so if you're moving things within a watershed, that is more practical, just to assume that the same pathogens occur within that watershed. But between watersheds, that's a long distance movement. And if you need to prioritize what you're doing, then that sort of, the longer the distance of the movement of something, the more issue with transmission of a pathogen that could cause disease could ensue. So there's other, like, large equipment considerations for fire and wildfire prevention practices as well. And I think a practicality clause there is if the equipment is getting into water, then there is more concern for those aquatic pathogens. You know, we haven't talked about snake fungal disease that is hitting some of the aquatic snakes, but it's also hitting terrestrial snakes. And so, you know, pathogens could be anywhere, but if you need to prioritize, if your heavy equipment is hitting an aquatic site, I think that is a higher priority.
And that's a good lead into heavy equipment question here for Paula. In general, how long does it take to brush down, disinfect, and rinse a large piece of equipment like a backhoe or a dozer? So I'm wondering if also, you know, maybe there's a dedicated team of folks that's their job to do, because it's going to take a lot of time.
It will take time, and that's part of the planning phase. In most cases, you have to clean your boat when you bring a boat out of water. So in this case, the same thing. You have to plan to be able to remove that mud caked in, and use high pressure air and water. In terms of disinfecting, that is from when I've talking with contractors, that is a little bit more of an issue than just getting the stuff off these vehicles if you plan ahead. So if you have an area where you can bring the vehicle, if you start from a non-contaminated area to a contaminated area, then you have a little bit of a grace period. But if you have a set site, sort of convenient to all your work sites, that you can bring your vehicle to, or your backhoe or whatever, and have everything there to rinse it out, then that, as I understand it, would be the optimal. I don't know exactly the amount of time though. Okay.
Hey, Paula, I have a follow-up question to you, or for Jim Julian, if he's still on. I've heard of field crews that will plan ahead for their work to be in a very small area for a week, for example. They work a 4-day week, and then they run their vehicle through a car wash and let it sit over the 3-day weekend before they use it again. How effective would that be for pathogens?
If the water is hot enough, and it has to be pretty hot, then I think it will deal with some of the pathogens. It generally, those truck washes have to be available, and they're not always available, but I know that we used them for some of the work that we did with surveys, and we took our regular trucks through them. So Jim, do you have anything to add?
I think our group did talk a little bit about that. I think one of the problems in applying that to some heavy equipment is, I mean, the massive amount of debris, you know, and sediment, and that, you know, some of these commercial car washes aren't really quite set up to handle that sort of, you know, that sort of demand. But I think for field, you know, regular, you know, sort of field vehicles, which DD, I'm not sure if that's what you were implying. I mean, that seems adequate.
Yeah, I think there's a spread of vehicles that people use, and so even your everyday, you know, vehicle, it might be a 4-wheel drive or some sort of small truck, a car wash, and then letting it sit for 3 days in the sun. I mean, that helps.
So we have a really important question here that hopefully we have time for one last question. So Erin works for the Corps of Engineers, and they do a lot of construction work. The question is whether there is any sort of legal requirement that would support the necessity of following biosecurity projects or practices on projects, because if in some cases, if folks were to try to incorporate these and require their employees to do these practices, they might be told that there weren't any funds available to follow those steps.
That's a great question and a great point. And I think that's the reason that we're trying to shift the mentality to making it part of the bidding process, a competitive bid where you are including it so that it benefits the person, the resources, and the person, the contracting, whoever is contracting you is getting that benefit. And again, it's a lot for a resource manager, it's a lot cheaper to take care of it ahead of time before you have to get rid of that invasive species or pathogen.
More and more, I'm seeing that people that work with wildlife, that they need to get state wildlife permits, or if they're doing research, they need to get an IACUC permit, and embedded in those permits are biosecurity requirements. So that's a small subset of people that would be using large equipment, of course, but we're starting to see the need for biosecurity tied to licensing and permitting. And so, you know, I think we're kind of at this tip of the iceberg where it's getting more and more recognized. For implementation for large equipment, I think it's when we can get it done practically at this point, I think we jump on that. And so the larger projects that are moving things larger distance, that might be an initial focus, but it's a tough issue right now.
Great. Well, thank you all. We're right at 4:30, which is our stopping time. Really, really appreciate our two speakers today, Dr. Katie Heyman and Dr. Paula Henry, as well as our other disease task team members who have helped jump in, like Dr. Dee Olson and Dr. Lisa Schender, who's been helping with all the questions and the chat. So it really takes a lot of people to help pull these things together, and we're really appreciative of all of this. So I'm posting a link again in the chat. That has all of our resources. We will provide the recording within a week or two. It'll depend on how long it takes to correct closed captions. But in the meantime, I've posted my email. So if you have any questions, you can reach out to me and I can send you the emails of any of our speakers, as well as for those of you who might need a certificate of attendance, then you can reach out to me and I can provide that. So thank you again, and I hope you'll consider joining in our future PARC conversations and look for our other webinars in the works. So have a great day.
Thanks, everyone.